Okay, we're here. We're going to talk about some special senses in the body today. The first that I have that I've drawn on the board are taste, and then also olfaction. Olfaction is referring to the sense of smell. We'll start over here with taste. So I have drawn a tongue. You can see our big tongue here, and then I have exaggerated. This is called a lingual papillae. There are 5 different types of lingual papillae, not super important. More importantly, we have taste buds. These are little indentations into the tongue, right adjacent to the lingual papillae. And then inside the taste buds, we have the gustatory cells. And the gustatory cells are where everything's going to happen. It's important to note that anything that we taste has to be dissolved in our saliva. That's one of our roles of saliva. It's to help dissolve food. That way we were able to taste it through our gustatory cells. A couple different things that we taste. We have 5: salty, sour, bitter, sweet, and umami. Umami is a flavor that's savory. It was discovered in Japan. That's why it's called umami. Umami is Japanese for savory. We'll start with salty. Salty and sour have their own. And then bitter, sweet, and umami all share a pathway. So I'll start with salt. Anytime you have something salty, you're going to have, say we have table salt, NaCl. It's going to dissociate in the saliva. You're going to get sodium. Sodium is going to come into what's called the gustatory cells through an ENaC channel. It's going to depolarize. The depolarization along the wall of the cell, membrane of the cell, is going to cause voltage-gated calcium channels to open. Calcium is going to come into the cell. It's going to activate our snare proteins. Snare proteins are going to pull the vesicles down here to help release this neurotransmitter, which will then go to the brain, and it will say, hey, we are eating something that has some salt on it. Second is sour. Sour is going to use the same ENaC channel. And sour is going to be a hydrogen ion. So anytime we have something sour in our mouth, once it's dissolved out, it's going to be the hydrogen ions that's coming down the ENaC channel. Going to depolarize the cell membrane again. Once it gets to the voltage-gated calcium channels, calcium's going to rush in. It's going to bind to those snare proteins, activate those snare proteins, pull the vesicle down to the membrane wall, and through exocytosis, it's going to release the neurotransmitter onto the cell. The postsynaptic cleft. It's going to go to the brain. Another thing that the hydrogen ion does is that it's going to block the potassium channel. The third is our bittersweet umami. These are going to be G protein complex receptors. So if you have anything that's bitter, sweet, or umami, it's going to bind to this GPCR. The alpha subunit is going to come off. It's going to activate IP3, which is going to go to the endoplasmic reticulum. You remember there's going to be a high concentration of calcium in the endoplasmic reticulum. IP3 is going to signal for the release of that calcium. Calcium is going to go out. It's going to activate these snare proteins once again on the vesicles. It's going to pull down the membrane wall and release its neurotransmitter onto the postsynaptic cleft, and then that action potential is going to go to the brain, and it's going to say, we have just eaten something bitter, sweet, or savory. That's taste in a nutshell. It's pretty simple, pretty straightforward. Another thing that's pretty straightforward is this olfaction. So our sense of smell, you can see here I have drawn a nose, a pretty rough nose. You'll notice, once again, this is similar to taste, is that our odorants, things that we're smelling, they need to be dissolved. Anytime it's an ion, it'll dissociate, and it will come up here, it'll dissolve in our mucus, and these we have our receptors here. They go up through this epithelial layer of the nose, through the cribriform plate, which is bony, and then it goes up into this olfactory nerve. And our olfactory nerve, we remember that's going to be a cranial nerve. It's going to synapse in the cranial nerve and go to the brain. It's going to tell us what we're smelling. So we'll look a little bit closer here. at these olfactory neurons, or olfactory cells. So we have an odorant that comes in. This is also going to be G protein coupled receptor. We're going to have our odorant bind here. Alpha subunit is going to come off. It's going to go activate adenylyl cyclase. Adenylyl cyclase is going to create cyclic AMP, which is going to open a cation channel here. We're going to have sodium, calcium, potassium. Cations are going to come in. They're going to depolarize the cell. Depolarization of the cell is going to cause this chloride channel to open up. With that chloride channel open up, the driving force for chloride is going to come out of the cell, which is going to further depolarize the cell. We don't know the synapse here. It's going to synapse up here on these cells, and it's going to go to the brain. So that's olfaction. Once again, taste and olfaction, pretty straightforward, pretty easy. And then we'll talk about sight and sound here in just a minute. Okay, so I have drawn on the board an eyeball. We're talking about focusing light right now. On the back of the eye, you have your retina. That's where all the light's going to converge. It's going to be able to interpret what we're seeing, and it's going to be able to send that message to the brain via the optic nerve. So I have drawn out light coming in. So the first thing that we run into is the cornea. The cornea is responsible for bending light, but it's just a set amount. It's always going to be the same. It can't change. So if we get closer or further away from something that we're looking at, that would be a problem. There may be a time when we're a certain distance where we just need our cornea, and it would be perfect. It would converge on the back of the retina, and it would make a good image. However, what we have on the inside of the eye, so is this lens that I've drawn out. And the lens, the good thing about the lens is that it is adjustable. So as we get closer or further away from something, our lens can relax or can retract based off of this muscle. This is the ciliary body, and it has these ligaments called suspensory ligaments that are going to the lens. And when that ciliary body contracts, as we get closer to something, it's going to pull the lens into a more conical shape, and it's going to help focus and help the light converge at the back of the retina. The further away we're looking at something, the ciliary muscles are going to relax. That lens is going to be flatter. Light's going to be bent from the cornea, not so much by the lens, and it's going to converge on the back of the eye. A lot of people, they'll have to wear prescription glasses because they'll be either nearsighted or farsighted. Nearsighted is myopia. That's when the lens is not able to converge the light enough. So you can see I have drawn down here that where it ends up converging is further back from the retina, so you're going to see not very clear. So what those people need to do, or what happens for these people, is they're going to give a prescription, and they're going to have a eyeglass with a conical shape, or a convex eyeglass, and that's going to help bring this in and converge on the back of the retina. We also have hyperopia, not as common, but still correctable. This is when the light converges before the back of the retina, so it actually crosses over. These people, what they need is called a concave lens. So it'll be thinner on the inside. And what that does is when that light comes in, it's going to push it out more. Pushes it out more, then when it gets to the lens will bring it back in, and it will converge on the back of the retina and be able to send a better picture to the brain. All right, we're talking phototransduction, rods and cones, on-center, off-center visual fields. we'll start off rods and cones. Rods and cones are the photoreceptors. They are in the back of the eye, right next to the retina. So if you think light's coming from that side in, you're going to come in, you're going to go through the ganglia cell, you're going to go through the bipolar cell, and then you're going to get to the photoreceptor cells. So the light has to filter through all these cells before it gets to the photoreceptor cells. In the back of the eye, like I said, we have rods and cone. Rods, they're going to be sensitive to light. Those are going to be good at night, good for night vision, increased visual fields. They're going to show us black and white picture. During the day, they're going to be bleached. Our cones are going to be the main thing that we're getting feedback from. Cones, not super sensitive to light. Definitely takes certain types of light to break these bonds down here. It's a little different molecule. I'll talk about that in just a minute. 3 different types of cones. There's red, green, and blue cones. Great for color vision, great for sharp vision. In the back of the eye, we have a depression in the retina called the fovea centralis. That's going to be where our cones are. Cones are going to have a little bit smaller receptive visual field. They're going to give us a sharper vision, more clarity. I have, these are all up here, these top 3, these are all photoreceptor cells. We just need to know the difference in what they do, but once we get into kind of the molecular breakdown of these cells, it's not too different. So we'll start up top. This is transducin. Transducin is activated by light, and I'll show how that's activated down here. Light comes in, then transducin, we have an 11-cis retinol, and what light does is it takes the 11-cis retinol, it breaks a double bond down there, and it makes it an all-trans retinol. If that happens, so we'll say light is here. Light is on this photoreceptor cell. Transducin, 11-cis retinol, has been changed to all-trans retinol. This is activated. So our alpha unit of our G protein is going to go down here. Phosphodiesterase. Phosphodiesterase is going to come to the cyclic GMP, and it's going to break it down. Breaking down cyclic GMP is going to close this cation channel. This cation channel was previously open, depolarizing the cell. We are no longer depolarizing the cell. We're no longer going to have voltage-gated calcium channels open. Calcium's not coming in. We have vesicles in here that contain glutamate. When light is on, we are going to have less glutamate coming down or being released from our photoreceptor cell. We'll come over here and we'll talk about dark. I'll show you the difference. Dark, we're not going to have the breakdown from 11-cis retinol to all-trans retinol. Transducin is not going to be active. Its alpha unit is not going to be active. We're not going to have phosphodiesterase. We're going to have phosphodiesterase disabling GMP. We are going to have cations coming in, depolarization of the cell, calcium ions coming in. Our snare proteins are going to be activated. We are going to pull these vesicles down to the bottom of the cell membrane. We're going to exocytose, and we're going to have a whole bunch of glutamate. So we are going to say we are going to have increased numbers of glutamate down here. So increased glutamate. Now you remember that it's not just the photoreceptor cells that are part of our visual cells. We also have the bipolar cells. There are 2 different kinds of bipolar cells. There's on center, off surround, and there's off surround, on center. Right now I have drawn on the board, this is the on center, off surround cell. So these are talking about our visual fields. This is how we are able to interpret lines, boundaries, patterns, and I hope that I can kind of explain that a little bit more, at least how this all works. So we have our photoreceptor cells, we have our on-center cells. So our center cell is going to be directly over our bipolar cell, and then our surround cells are going to be off to the side. We also have another cell in there that's playing a role. It's called our horizontal cells. Our horizontal cells have a cell receptor called AMPA on them. AMPA is sensitive to glutamate, so if glutamate comes in and binds to AMPA, we're going to have depolarization. So we have glutamate binding to AMPA, and we have calcium coming in, we have depolarization, calcium coming in, and we have release of GABA. So if we have increased GABA, GABA is interesting because it's going to inhibit, it's going to be a neuromodulator for our photoreceptor cell, our center photoreceptor cell. So I'm going to draw that really quick here. We are going to have GABA come up. It's going to bind here. This channel is going to open. We're going to have either chloride, run into the cell, hyperpolarizing it, or we're going to have potassium run out of the cell, also hyperpolarizing it. Either way, we have it come into here, and we are going to get less glutamate. We're going to have less glutamate coming off of that cell because this photoreceptor in dark is producing more glutamate, more GABA. The GABA is inhibiting the glutamate from the center photoreceptor cell, which means the center photoreceptor cell is going to release less glutamate. And I've drawn it where there's these dotted lines here because it all comes, it's all added together. So it's all summed together, and we're going to get a percentage of an action potential down here that's going to go to the brain. So we've worked through that one. Let's finish working through this one on the light. So less glutamate, AMPA's going to be going to not react as much because there's less glutamate. So we're going to have a little bit in here. We'll have less. We'll just draw it here. Less glutamate here. Or less GABA. Excuse me. Less GABA. Less GABA. Less reaction here. Less reaction here. More glutamate here. All right. Now we can look at our center cell. And let's put some dark on it. Dark. So dark, you'll remember, nothing's happening. Our 11-cis retinol is not going to be converted to all trans retinol. PD is not going to be activated. Cyclic GMP is not going to be broken apart. We have a cation coming into the cation channel here. Calcium's coming into the voltage gated channel here. We are going to have snare proteins activated. Vesicles are going to be pulled down to the bottom of the membrane. And we are going to have increased amounts of glutamate here. So we can think of it kind of separate. We have, overall, we have an increased glutamate from this cell over here. We have an increase in glutamate from our center cell over here, and a decrease in glutamate from that cell over there through everything that we've done. We're going to give them, we're going to say it's 100%. We're going to give this one 60%, so we're just going to call it 6, 2, and 2. And these are the potential, action potentials that are going to go through the ganglion to the optic nerve into the brain. So there we have it. We're going to talk about mGlu-R6. mGlu-R6, that's going to be for a on-center receptor. So when mGlu-R6 is not active, we have cations coming through this TRMP1 channel and depolarizing the cell. That's sending vesicles, neurotransmitters that are excitatory to the brain. However, when mGlu-R6 is activated, when TLR6 is activated, it will bind this TRMP1 channel, and it will close it. And it will close this voltage gauge calcium channel because we're not getting depolarized anymore. So with increased glutamate from the photoreceptor cell onto the bipolar cell that it has an mGluR6 or is an on center, we are going to have less release of neurotransmitter, less action potentials going to the brain. So let's come up here. We remember from our photoreceptor cell, we had increased glutamate. So if we have increased glutamate up there, we're going to have decreased glutamate down here. Decreased glutamate down here. Our middle one, we have increase. We're going to have decrease again. Decreased glutamate down here. Decreased, let's say action potential actually. Yeah, action potential. Decreased action potential down here. Less glutamate is going to keep this inactive. We're going to have cations coming in, depolarizing the cell, vesicles coming out, neurotransmitters into the intersynaptic cleft. Action potentials are going to increase. Increased action potentials. Okay, so if we look at the total summation of all of these interactions in the cell, we're going to find that we're going to have 0 for this side, 0 for the middle, 2. So 20%. We're going to have 2 action potentials going into the ganglion, and this is going to the brain. And this is how we get, like I said, this is how we were able to tell boundaries, lines, patterns. This was an on-center bipolar neuron. We also have off-center bipolar neurons. And I'm going to show you the difference. Okay, we're on our bipolar neuron. We are not changing anything on our photoreceptors. So here we go. We're going to start down here. This is going to be AMPA. And AMPA is not a G protein receptor. AMPA is a channel. So we'll just draw AMPA there. We'll draw a little channel right here. AMPA. And AMPA, when AMPA is open, we are going to have TRMP1 is not on the channel, so we're going to just load it. AMPA's a channel. Glutamate is going to stimulate AMPA to open. AMPA's open, it's going to cause depolarization. There's going to be cations coming in here. Depolarization, you're going to have calcium coming in here. Vesicles containing our neurotransmitter. Snare proteins are going to be activated. They're going to pull the vesicle through the cell membrane. We're going to have depolarization. We're going to have increased action potentials. So let's look up here. Let's see how it's different. So if we have increased glutamate on our AMPA receptor, we're going to have this channel that's going to be open more. If that's open more, it's going to depolarize. We're going to have calcium coming in through the voltage-gated calcium channels, vesicles getting pulled to the membrane. We're going to have increased action potentials with AMPA coming from this surround. From the middle, from the center, we're going to have increased glutamate, so we're also going to have increased action potentials. We're going to have decreased glutamate, we're going to have decreased action potentials. We'll assign these numbers 2, 6, 0, because we are not getting that action potential with a decreased amount of glutamate. Total's going to be 8. We're getting 80% of our action potentials from our off center on surround with light on one surround and dark on one surround and the center. All right, I hope that makes sense. I hope that you were able to understand it. Let me get this erased and then we'll talk about hearing. Okay, let's talk about sound and hearing. Sound, if you think about it, sound, when we produce sound, we're creating waves, sound waves that go through the air. A couple different things we'll talk about with sound waves. Sound waves have frequencies, so you can have a higher pitched sound that's going to have a higher frequency. The wavelengths are going to be shorter and more frequent. Lower sound is going to have a lower frequency. Longer wavelengths, less frequent amplitude. That's just talking about the loudness. It's measured in decibels. Yeah, with those two understandings, we can just talk about the ear. So if we have a sound wave, so someone's talking to us, in our external ear, so we have our external ear, our middle ear, that's from our tympanic membrane to the cochlea, so middle ear, and then our inner ear. So our pinna on the outside of the ear catches that sound. The sound comes in here, and it vibrates the tympanic membrane. The tympanic membrane is connected right here to the stapes, or to the malleus. The malleus vibrates. That vibrates the incus. That vibrates the stapes. The stapes is on the oval window here. And that's the end of the cochlea, and the oval window is connected to the scala vestibuli. So we'll just call this the scala vestibuli, SC, and it goes up, pushing this now fluid. And once it starts coming back, it's called the scala tympani. So we'll just call this ST, scala tympani. We have a round window here, because if you put force into something, you have to have give somewhere. So the round window is going to allow it to give and not break anything. The important thing here is what we have in between here. So this right here is our basilar membrane. And our basilar membrane is what contains the hair cells that read the vibrations and are able to interpret sound and send it to the brain. I have a cross section of this, so if we cut it open right there, I have a cross section drawn down here. Scala, what did I say that was? Scala vestibuli. I don't know why. I have an SC, should be an SV. Scala vestibuli. Scala tympani, cochlear duct in the middle. Then we have our hair cells right here on our organ of Cordy. Organ of Cordy. And these are on the basilar membrane. If you've ever played the xylophone, I like how it explained it in the book. There's different lengths. Each length has a resonance. So this basilar membrane, it's not straight, but it looks something like this, where it has an increase in length. So if you play something high pitched, it's going to be like on a guitar, it's going to be tighter. The string is going to be shorter. It's going to resonate on this basilar membrane somewhere up here. This is where we're going to get the vibrations. And the rest of the membrane, it's just going to pass by. It's not going to vibrate it. If you have something that's low, think of those long frequency waves, that's going to resonate somewhere down here on this basilar membrane. So think about that when we're explaining all of this with the hair cells. Another important thing that we have to set up is some gradients here. So we have perilymph and endolymph. Perilymph is going to be down in the scala vestibuli, so we'll just say peri here, and also in the scala tympani, so we'll say peri here. And this is actually going to be endolymph here, endolymph in the cochlear duct. And I'll show you some concentrations. The concentration of potassium is huge in the cochlear duct, and I'll show you how it gets that way. You see I've drawn these green cells outside of here. These cells we're going to call the stria vestibulae. Stria vascularis, let me get that right. So the stria vascularis, what it does is it its goal is to push potassium into the endolymph. So we have sodium potassium pumps, we have these NAKCl channels that push, or pumps that push sodium, potassium, and two chlorides in there. We have a chloride channel that allows chloride to come back out, and then we have potassium leak channels over on the other side. We have another of these same type of cells here. It does the same thing. We have a sodium potassium pump, we have a NAKCl, and then we have a Cl channel, as well as, again, these potassium pumps on the other side. So our potassium concentration is close to 150 milliosmolar in this cochlear duct. So if we think about that, we have the endolymph up here, we have 150 milliosmolar. And then the perilymph, we do not have 150 milliosmolar. In fact, we have a lot less. In the cell, we have about 150 milliosmolar as well of potassium. The big driver here for potassium to go down to this hair cell when it vibrates is an electrical gradient that is formed because we have such a positive gradient here, an even lesser gradient in the hair cell, and an even less charge out here in the perilymph. So what happens is we have sound. It comes in through our ear, vibrates in the tympanic membrane, malleus, incus, stapes. It sends this wave in the scala vestibuli. So we have this wave, and somewhere along here, it finds its not frequency. It finds its resonance. So once we get to that length of the basilar membrane that has a resonance for, it's going to vibrate. When that vibrates, this hair cell is going to vibrate and wiggle, and you notice how it has these projections on top, microcelia? I'm not quite sure. But when they vibrate, they have these linked potassium channels. When they vibrate, they'll pull, and it'll pull open some of these gates on the potassium channels. Potassium from the endolymph, remember, it's so much, but the big thing is the electrical gradient. So we're going to have potassium getting pulled in to this hair cell. It's going to cause depolarization. Depolarization, we'll have voltage calcium coming in. It'll be activating our snare proteins, pulling our vesicles through the cell membrane, and we're going to get an action potential going down the vestibular nerve into the brain, and we are going to interpret sound. That pretty much covers it. We should be good for next time.